Tonight's speaker, Dr. Nicholas Mason, is an assistant professor in LSU's Department of Biological Science, where he's teaching ornithology. And he is also the curator of ornithology for LSU's Museum of Natural Science. Dr. Mason came to LSU in 2020 following Van Remsen's retirement. His educational background is impressive. After receiving a Bachelor of Arts in Honors in Biology from Vassar, he went on to receive a master's in biology and evolutionary biology from San Diego State University. And then he received a doctoral degree in ecology and evolutionary biology from Cornell under the direction of Irby Lovett. Dr. Mason's research centers on ecological and evolutionary processes that drive diversification in birds. He and his students also study how birds respond to human activities in hopes of mitigating the loss of biodiversity in our rapidly changing planet. Dr. Mason's lab and all of the research projects use natural history collections in their studies. He and his lab undertake local and international field work to study avian biodiversity via scientific collecting. He and his students are dedicated to the continued growth and stewardship of natural history collecting. Dr. Mason will now give us an update on the ornithology program and the world-renowned bird collection at the LSU Museum of Natural Science. So Dr. Mason, I turn this over to you now. Thank you.
Wonderful. Thank you so much, Jennifer, for the introduction. So I'm going to share my screen. Okay, can everyone see that okay? Is that coming through? Okay, excellent. All right, yeah, thank you, Jennifer, for the intro, and thank you for the opportunity to speak to all of you. It's a shame we couldn't do this talk in person, but it seems like things are looking up with Omicron, and so I'm hopeful the next time I have the opportunity to speak with all of you that will be in person. I look forward to that day and to meeting all of you then. So as Jennifer said, I wanted to present sort of a holistic synthesis and my vision or idea of kind of what we do here at the LSU Museum of Natural Science. So I've titled this Looking Backwards, Forwards, and Up. And so some of this will be a retrospective. I'm going to talk a little about the history of the museum. It's got a very interesting history in terms of the people involved and the timeline into where it is today. Also looking a little bit ahead. Right now is actually a very exciting time for the museum. There are some big changes that are forthcoming. And then in addition to being ornithologists, we're also birders. And so we're also always looking up to the trees and thinking about birds as well. So I'm going to present some of our work that we do on birds and the different projects that we're currently undertaking. And I'll just start by saying, so I'm not a Louisiana native. You can probably tell that by my accent, but I'm really enjoying getting to know Louisiana and the state more and more. Not just with the birds, but with the culture. This will be my first in-person Mardi Gras. I'm thrilled for that. So if you have any recommendations for things to do during Mardi Gras, I'm all ears. Please send them to me in the chat or via email. So I thought I'd start off by thinking about, so what is a museum anyways, right? So when you hear the word museum, you may conjure an image of something like this. Maybe a museum to you is a collection of interesting things or a place where you can go to think and contemplate, or maybe it's a place where culture is preserved. And it's also important to think about who has who benefits from the museum and who has access to the museum. And I think in the distant past in Greek culture, this used to be something that was reserved for people that only had certain prerequisites for appreciating certain things like poetics or music or philosophy. But I'd like to dispel that and talk a little bit more about what the museum is here that we have at LSU. So what are the different things that we do as an institution? Probably the primary thing that we do is we generate new knowledge. And so we undertake different studies in ornithology focused on trying to understand the natural world and specifically about birds. And so I'm going to talk a lot about different examples in which we've done that. One of the most prominent ones recently is this phylogeny here. So this is a phylogeny that depicts evolutionary relationships of a group of birds that are called subausines. It's a large radiation of birds. And people here at LSU have been spending decades trying to understand the evolutionary relationships among thousands of species. And so this is a big undertaking. This was just published recently. We also collect and preserve specimens as a record of Earth's history. And so when many people hear natural history collections, they may think of the days of John James Audubon and other people who would go and collect specimens, and maybe they think of these specimens that have just been collecting dust in a drawer. But active scientific collecting is something that we still do and we care deeply about as a way to try to understand how the world is changing. And so we also are active in terms of a scientific collecting institution. We educate both graduate and undergraduate students and attempt to train the next generation of biologists. And so we recently graduated some of our graduate students. So this is Oscar Johnson, who's now doing a postdoc at the University of Texas, El Paso, and Jesse Salter, who's now doing a postdoc at the Natural History Museum in Los Angeles. And so we're very proud of the students that go on to do really amazing things after their time with us. And then we also attempt to educate general public via exhibits and outreach. And so that's one reason why I'm excited to have this opportunity to speak with y'all. We all think about ecological and evolutionary literacy in the general public as something that we try to promote. And so we do that through exhibitions that have been a big part of the museum. And if you've been to the museum, hopefully sometime in the past or in the near future, you'll get to see these really beautiful dioramas that we have on our public side. But we're also involved in outreach with things like local interest groups like the Audubon Society and master naturalist groups and things like that to try to encourage a deeper understanding and appreciation of nature. And interestingly, we also assist with local parties with identification and consultation. So just recently there was a bunch of illegally confiscated things that were illegally procured. And so one of the things that we do sometimes is provide assistance to government entities in terms of identification, which can actually then be used in court cases. So we don't need to do this too often, but it is still a service that we provide as a museum. We have this repository of specimens that we can use to ensure correct IDs. So here's a brief outline of what I'd like to talk about today. And this is similar to a talk I gave for the Baton Rouge Audubon Society. My parents watched that and said, man, you put too much in there. So there's a lot in here, but I'm excited to share this kind of holistic vision of what we have going on at the museum. And so these are all the different things I'd like to talk about. I'm going to start with a really brief history of the bird collection, which is actually quite fascinating and something that I only really recently dove into. So the collection was founded in 1936 by this gentleman, Dr. George Lowry. He was actually born in Monroe, Louisiana, so he was born and raised in Louisiana. He did his PhD at Kansas, but then came back to establish the museum in 1936. Public exhibit was then opened in 1955. And George Lowry was a fantastic naturalist, so he's not just an ornithologist, but actually learned and studied a lot of the mammals of Louisiana as well. So some of the things that Lowry was known for was this method of observing the moon to try to count and track migratory birds through spring and fall migration and estimating the abundance of migratory birds. But he also published some really important seminal works on nature in Louisiana, including Louisiana Birds in 1955 and the Mammals of Louisiana and its adjacent waters in 1974. And there's a really interesting book. Let's see if I can, I'm going to try to put this in the chat. We'll see if this works. So I'm going to put a link in the chat. Yeah, okay, I think that's working. So there's a link in the chat there that I just put down, which is a PDF that we have on our website. And if you're interested in learning more about the history of the museum, I encourage you to check this out. This is a really interesting account by someone named Peter Soderbergh, who wrote this book called Windows on the World. And it's all about the public exhibits in the museum, but also the early days of the Natural History Museum. And so it's a really fascinating account of kind of the beginning of the Natural History Museum. But we really think of this museum, and not just me, I'm biased because I'm the curator, but I think, you know, across the university, it's really a special thing that we have, this institution. It's the third largest university affiliated collection in the world. It's a really actively growing community, and it's a special resource we have for understanding our natural world. And so we're very proud of our public exhibits and also our research, and that's shared across the university, which is really great. So George Lowry, unfortunately, had an untimely death. He suffered a heart attack, and so he died suddenly. And in 1978, his student, John O'Neill, became the new curator. And so he was the director of the Louisiana State Museum of Natural Science after that. And he was also an extremely talented painter. And so if you come to the public exhibits, you'll see his fantastic paintings throughout the institution. They're really beautiful. He also described numerous species that were new to science. One of them is this orange-throated tanager, one of my favorites. Does this bright orange throat. It's from a small region in Peru. It was described by Lowry and John O'Neill in 1964. And John is actually having his 80th birthday, so we're going to hopefully throw a big party for him later this year. It should be fun. So one of the really prominent figures in the history of the Museum of Natural Science here at LSU was Dr. Ted Parker III. And so Ted Parker was an incredibly gifted naturalist and ornithologist. Many describe him as the best field ornithologist that they've ever encountered in the 20th century. He pioneered the use of playback, and so he was one of the people that really thought about using audio recordings in order to draw in birds. He would record birds and then draw them in. And he was so talented with identifying birds by ear. There are stories where he would identify new species by ear and say, well, that's different, that's a new species, and then he turned out to be correct. So he was an incredible naturalist. He unfortunately died in a plane crash in 1993 with botanist Al Gentry. And so he was a research associate at LSU, but is really, really much, much more. He's really an inspiration for many of us here. He dedicated his entire life to trying to understand neotropical birds, and is really an inspirational figure that we have here at the museum. And there was a really excellent book called A Parrot Without a Name. If you're interested in tropical ornithology and want to learn more about the LSU Museum, there's an entire book written about expeditions. And it's actually a really fun read too. And there's many of the people in it are still alive and still active in ornithology today. I'll put another link in the chat. Hope that's okay. But this is a link to Sora, which is it's an ornithological archive, and there was a special edition after Ted Parker's death that was written, and there's a really nice in memoriam that's written about Ted Parker. So if you want to learn more about this pretty amazing individual, the first introduction that's written by Van Remsen is really worth checking out. It's a short read, but it's pretty remarkable how impressive Ted Parker was. So then, you all are probably familiar with Van Remsen. So Van Remsen was my predecessor, and he was the curator of the collection from 1979 to 2019, an expert naturalist, contributed extensively to our understanding of both local as well as South American birds. He established basically what we have for classification and taxonomy in South America, but is also an authority on Louisiana birds, and had a really nice article recently on the importance of Louisiana birds in Wilson Ornithological Society in their journal. And he, along with Mark Hafner, who was the curator of mammals, established the Genomic Resources Collection here at LSU in 1980 and 81. And we actually have the largest genomic resource collection in the world. And so, little LSU on the bayou has world prominence in terms of the foresight that some of the ornithologists and mammalogists had in terms of thinking about DNA as this important resource for understanding the natural world. So that's really exciting. And so I have huge shoes to fill as Van's successor, but Van is still active in Louisiana ornithology and a great resource and a great guide. Bob Zink was another curator that actually moved on, but he was at LSU from 1984 to 1993 and was a pioneer in understanding molecular systematics, so using DNA to understand relationships among species, and really helped to grow the genetic resource collection. He was actually the first curator of genomic resources. And we actually have an ongoing faculty search right now at LSU for a new genomic resources position. So this is sort of emblematic of how LSU was sort of on the front foot in terms of thinking about this aspect of evolutionary biology and ecology. You probably also know Donna Dittman and Steve Cardiff, fantastic curators, fantastic people. So they've been a huge part of the LSU Museum for the past 30 years, and they've helped the museum grow immensely and have trained countless people in terms of specimen collection and preparation, and have been leaders locally in the Ornithological Society. And just a shout out to the Yellow Rice and Rails Festival. I got to go this year. It was so much fun. So if you've never done that, I highly encourage you to check that out. And then just a couple more folks that I want to point out. So Fred Sheldon is actually retiring this semester. He's giving what he's calling his exit seminar on Friday. So this is his final semester with the museum, but again, he was someone who really pushed the envelope in terms of thinking about DNA as a resource for understanding evolutionary relationships among birds, or in other words, systematics, and really expanded the geographic scope of LSU to include all the work that he's done in Southeast Asia, in Indonesia, Sulawesi, and other parts of the Malay Archipelago. And last but not least, Rob Brumfield, also originally from LSU, came back as a curator in 2003, and again emphasizes our strength in speciation and systematics. And he was behind that giant phylogeny that I showed. He's been really at the forefront of systematics for decades now, and is now moving up in the college world. He's an associate dean in the College of Sciences, and still maintains his position here at the museum as well. And so many of the people that have been at LSU have gone on to hold prestigious positions at museums all over the world, really. And so that's something that we're really excited about. These are just some of the people that have graduated from LSU and gone on to hold positions. So Shannon Hackett at the Field Museum, along with John Bates, Chris Witt at the University of New Mexico, Terry Chester at Smithsonian, my master's advisor Kevin Burns, and on and on and on. These are many of the most prominent museum-based ornithologists in the world, so we're extremely proud of our alumni as well. And what's really cool about LSU is there's a really active ornithology community beyond the museum as well. And so in addition to the museum, there's folks like Phil Stouffer, Jessica Eberhard, Mike Polito works on penguins in Antarctica, Kevin Ringelman, who works with ducks and duck management and renewable natural resources, Sabrina Taylor, she works with seaside sparrows and other endangered species, Ashley Long, who's an assistant professor as well, works on spatial ecology of different birds. There's many, many ornithologists, makes this an incredibly exciting and enriching community for those interested in birds. And just a shout out to my non-ornithologist colleagues too. What's cool about the museum is we have specialists in other taxa as well. And so we have an anthropologist, an ichthyologist, geologist, mammologist, and a herpetologist all sort of in one unit. And there's lots of different challenges in terms of thinking about a museum and the different aspects of the museum for these different taxa and different aspects of natural history. But it's a really fun, exciting place to be for this. All right, so a little bit on sort of our collection. So kind of an update on where we are now in terms of numbers. So we are one of the most active natural history collections in the world, and that's especially true since 1980. And so this graph here shows an accumulation of specimens, different types of specimens. And you can see there's a very sharp curve here at the beginning, and it sort of starts to slow down a bit. But then in 1980, you can see this really sharp uptick in the number of study skins and also in the number of tissues. And so genomic resources really started to take off then. And so we have lots and lots of more recent specimens. And with those more recent specimens, they carry tons of data. So they have all of this information related to the breeding condition, related to their molt and other aspects of their biology that many of the older specimens lack. So these are really, really valuable specimens that we have. We have more than 220,000 bird specimens. That includes 175,000 skins, 25,000 skeletons, and many fluid specimens. So we're the third largest university-based collection in the world, and we continue to grow. So here's a global distribution of our specimens. You can see that we have in these hexagons, the more redder the color, the higher number of specimens. So you can see our global distribution here. We have a lot of birds from South America, but we also, Southeast Asia, so Fred Sheldon and his group have worked here. Fewer from Africa, but some, and then quite a few from North America as well. That's sort of just a snapshot. But I'd like to zoom in particular to Peru and Bolivia. And so as you may well know, LSU has had a really long history of working in these two countries, which are some of the most diverse countries in the world in terms of their birds because of this remarkable elevational gradient across the central Andes. And so LSU has had a really long history in working in these areas. And this is just another heat plot showing, so the darker the purple, the higher the number of specimens. So you can see the most specimens that we have are actually from these two regions, which are in the lowlands in Amazonia. So we have fewer specimens from the highlands of Bolivia and Peru. And that's actually one of the things that I'm hoping to focus on during the first part of my time here. One of the other things we're also really proud of is that we've contributed a lot of new knowledge by describing new species. And so as of now, there's been 46 new species that have been described by LSU personnel. These include some of my favorites, like the long-whiskered owlet, Xenoglox lowryi, the Inca wren up here in the top left, black-faced cotinga, lots and lots of really fascinating, cool birds from mostly South America that have been described, but also Southeast Asia as well. And this is still going on. So just recently you may have heard, but there was a new species described by LSU personnel last year. So Dan Lane is a research associate here. He's a phenomenal birder. I'm sure some of you have probably heard of him or encountered him during your birding adventures in this state. He's a phenomenal birder. And he and a number of other collaborators led the charge to describe this species of bird, which is called the Inti tanager. So Inti is the Quechua or the native word for sun. And so it's a very sunny yellow bird, but was unknown to ornithology largely because it's an intratropical migrant. And so it breeds in the deciduous forest in the raiding season in this very, very small part of Bolivia, and then migrates to this sort of more lowland area of bamboo in Peru when it's not breeding during the dry season. And so it managed to escape notice until just recently, but again, we're very, very proud of this contribution that we've done to ornithology. All right, so that's just a brief history of the collection, a little bit about the current state of the collection. I'm now going to talk a little bit about some of the ongoing work that's going on here. So I'm going to showcase, basically I asked the graduate students to give me one slide that summarizes their current research projects. So I'm going to share those with you, and then I'm going to talk a little bit about my own lab group and the projects that we have going on with my lab group specifically. But these are grad students that are outside of my lab group but are still ornithologists working with the museum. So first we have Diego Cueva, who is originally from Colombia and recently started working on this mannequin hybrid zones. This was a group of birds that Rob Brumfield actually worked on, I believe when he was a grad student, and they have a hybrid zone where they have the white-bearded and the yellow-collared mannequin come into contact. And so Diego's expanding this work to focus on a hybrid zone in the Chocó region of Colombia, which has not been very well studied, partially because of political reasons in the past made it difficult to access, but he's excited to sort of expand our understanding of the dynamics of this hybrid zone, which has kind of served as a model for studying other avian hybrid zones. Sticking with hybrid zones, we have Eamon Corbett. And so Eamon studies grackles, and specifically he's focused on the hybridization between boat-tailed and great-tailed grackled. One of the interesting things is he's actually finding that despite their similarity in appearance, they're actually not hybridizing as much as we originally thought. But he is studying them throughout their range, including here locally in Louisiana, to try to understand not just hybridization, but the genetics of eye color. And so one of the major field marks of these birds is differences in the color of the pupil. And so he's been collecting specimens specifically to try to understand what parts of the genome are involved in controlling eye color. Okay, so Anna Hiller is a grad student in Rob Brumfield and Brand Faircloth's lab, and she's studying these really cool flower piercer birds. So if you're not familiar with flower piercers, they're tanagers, and there's this complex in the northern and central Andes where there's hybridization between these different forms, and actually have what's called a leapfrog pattern of plumage. So you can see these black forms are sort of interspersed between these ones that have a rufusy chestnut belly. And so she's interested in generally why there are so many species of birds in high elevations, and is using this system to try to look at specifically what's going on in terms of hybridization dynamics and how does that play out with this interesting pattern of plumage across geographic space. Alberta Canton is our next student, and she's in the Faircloth lab, and she studies these mannequins that have difference in the color of their crown. And so she is using different types of spectroscopy to look at differences in the feather structure and what controls these iridescent colors at sort of the scale of microns and in nanostructures. And so there's differences from this electric blue to white to more sky blue, and she's quantifying differences in the morphology of the feathers at a very fine scale to try to understand what's driving that. And she is collaborating and building off of work pioneered by Andre Moncrief, who's also studying mannequins, but in more of a geographic, sort of phylogeographic context. And so there's this interesting group of Lepidothrix mannequins, the blue-crowned mannequins. It's this really widespread species that occurs across the Amazon basin and beyond. And so it's a great system for understanding the role of riverine barriers and other biogeographic barriers in shaping diversity in this group of birds. Marco Rego is a Brazilian ornithologist, and he's interested in using museum specimen records. And so you'll see it'll cycle through some different complexes here. And in these maps, these different colors correspond to different subspecies distributions. And so you can get really fine-scale, detailed information on where these different subspecies occur and how that translates to turnover. And so on this map in the bottom right here, these bright yellow colors indicate where there's lots of turnover. And so in the Andes Mountains, there's changes in distributions over short geographic scales, as well as in the Amazonian basin. And so this helps identify gaps in our understanding and knowledge of Amazonian bird distributions. And then his partner Glaucia is also interested in Amazonian diversity, but she studies hybrid zones and speciation, specifically in this group of birds called antbirds. And so there's a Regmatorrhina genus that has these two different species of antbirds, and they hybridize in certain parts of the Amazonian basin. And she's studying these different aspects of how speciation occurs. And so there's some kind of jargony big words here, but she's interested in understanding dynamics of how different traits go across hybrid zones and incompatibilities at the genetic level. So if you have two different lineages that hybridize in nature, maybe there are aspects of their genome or their DNA that are incompatible. And she's interested in understanding how that contributes to the speciation process. Dr. Oscar Johnson just recently finished his PhD with us and has now moved on to a postdoc, but he's been involved with the study of these subausine birds and has collected a large data set on different aspects of the biology, from morphometrics, so things like wing length or tail length, to differences in plumage coloration and song, and then trying to use all those data to understand geographic distributions. And so the question being, once a lineage diverges and speciates, and so there's two lineages, how long does it take for them to come back into contact? And what does that mean for their phenotypes that are involved, or their appearances? So he is studying the sort of macroevolutionary diversification of this group of birds. Rafael Marcondes recently finished, and he studied this group of antbirds that vary in their plumage. So there's a variable antbird, and found this gene that has been repeatedly implicated in vertebrate genomics. And it's kind of one of the exciting things right now is we can sequence whole genomes of these organisms, right? And so that was impossible 5-10 years ago, and has really transformed the kind of things we can do. But now studies like what Raphael led are able to identify specific genes. And so in this case, it's called the agouti signaling protein gene, but it's the same gene that controls coat color in rats and in mice, is responsible for being for basically an entirely dark black plumage to one that has white as well as black. And so he's able to identify the genes important in phenotypic diversification. All right, so that was sort of a showcase of different projects that are ongoing in the museum. I'm going to talk a little bit about specifically work that I do with my lab group and that I've done in the past, as to try to kind of give you an orientation to the kind of topics and questions that I'm interested in. So as Jennifer said, I'm relatively new at LSU. I started here in summer 2020, in the middle of the pandemic, and it's been great to build a lab group. And so currently we have 4 members of our lab: David, Subir, Samantha, and Maggie. I'll talk a little bit about their interests, and then a little bit more about kind of the ongoing work that we have. So Samantha is really interested in structural color and iridescence specifically. And so she's landed on starlings as her study system, which are a really fantastic, beautiful group. So we have our European starling here, of course, but Sternidae, and maybe those of you that are going to Ghana will get to experience some of these, hopefully in person, but they're beautiful, beautiful iridescent colors are really interesting from both perspective of what produces them at the molecular level, and then how that relates to their ecology and their evolution as well. In the past, she's worked on brood parasitism and thinking about cowbirds and visual systems and how hosts are able to reject or accept eggs in their nest from brood parasites. David Vanderplumm is a second-year PhD student. He's interested in altitudinal migration. And so we're in a migratory flyway here in Louisiana, but that is one that is a latitudinal flyway, right? And so we have birds that fly north to breed, and then will fly south in the fall during their non-breeding distributions. But what's interesting is there are many birds, especially in the tropics, that undertake migration across elevational distributions. And so they'll actually go up to higher elevations to breed, and then will return to lower elevations during non-breeding seasons. And so David's interested in studying this phenomenon and how it relates to speciation. And so maybe if you have differences within a species in terms of which populations migrate or don't across elevational gradients, that could influence whether or not they exchange genes and whether or not they become different species. So he's studying that in the slate-throated redstart, Myoboris miniatus. I don't know if that's actually occurred in Louisiana, but it does sometimes show up in Texas, I think, and other parts of the South. Maggie Mcpherson is a postdoc in my lab. Oops, one of her pictures didn't show up here, but she's interested in a variety of things, including she's done work with great-tailed grackles and their behavior. So she's done a lot of work behaviorally, thinking about decision-making in great-tailed grackles. More recently, she and I have been working with Tyrannidae, so the flycatchers, and thinking about the evolution of migration in this group and how that might relate to the morphology, and also how changes in migratory behavior occur. And so how a species goes from being sedentary to a migratory species. And Subir is the other member of our lab group. So here he is working hard in Southeast Asia, preparing specimens. And this is, I think, a slide showing some of his journey. So he's actually originally from Nepal, but he joined LSU via Arkansas and has been a big part of the, so he did his PhD at LSU with Fred Sheldon and has now joined my lab as a postdoc. But he's really interested in the biogeography of Southeast Asia, as well as the genetics of coloration. And so these are some of the species that he's worked on. He's actually a really good artist as well. And so each of these were drawings that he made. Okay, so briefly I want to talk about my lab sort of writ whole, or writ large. And so I've shown you some specific examples of work that other folks have done. I'm going to talk just briefly for the rest of the time about some projects that I've done. And again, this is all sort of snapshots. I'm just going to talk very briefly about it. So if you have any questions about the specifics of projects or you want to learn more, please feel free to drop questions in the chat or ask me after or shoot me an email. I'm happy to chat about these projects anytime. But broadly, so I consider myself a biodiversity scientist and a museum-based ornithologist. I'm interested in what drives the generation of diversity, both in terms of species, but also in terms of phenotypic diversity. So why are there so many species of birds? Why do they look the way they do? Why do they occur where they occur? I'm also interested in how we can try to better maintain biodiversity. And so we have a rapidly changing planet, and what can we do to try to promote the preservation of biodiversity or understand how birds are reacting to the changes that are occurring in their environment. So I'm going to focus really briefly on these 6 topics. So these are the things that I would say define my lab group. And I'm going to show some brief examples of projects that I've done that kind of touch on these different ideas and explain sort of what we try to understand through these different core concepts that really sort of define my interest in ornithology. So one of the things that I'm interested in is understanding speciation in systematics and taxonomy. And so you may have heard me say these terms already, but just to define them, systematics is sort of the branch of biology, trying to understand relationships among organisms, as well as diversification. And so that's like the evolutionary history of a different group of birds. And then taxonomy is our classification system that we use for understanding nature. So I'll talk a little bit about some different projects that I've been involved in. And one of those is with redpolls. And so this is a project that I did in collaboration with some folks and friends at CU Boulder. You may know redpolls as this group of 3 species currently that are distributed throughout the whole Arctic, and so northern North America, but as well as northern Europe. And they differ in terms of phenotype, but what we found is that these are really kind of a continuous gradient in their appearance. And you may have experienced that if you go to the far north and spend time in the cold trying to pick out a hoary redpoll from a flock of common redpolls. They look pretty similar sometimes, and sometimes there's intermediates, and we don't necessarily know how to classify them. But what we did is we actually sequenced DNA from these things, and what we found is that they're pretty much indistinguishable using DNA. And so what we suggested was that these are probably better treated as a single species, much to the chagrin of a lot of listers that didn't want to lose a tick off their life list, understandably. But so in terms of the evolutionary history, it seems like these are populations that are actively exchanging genes, and the phenotypic differences are probably not so much representative of different lineages of birds, but are more maybe related to local adaptation and environmental conditions throughout their range. So we had a paper that recently came out expanding on this work. I'm happy to share the link if you're interested, but we'll probably be proposing a lump of the redpolls in the nearest future. But it's not all bad news in terms of life lists. So another group that I've been working on are the Stellar's jays, and so this is a project I've been working on with some friends in California. Carlos Cicero is the staff curator at the Museum of Biodiversity, where I did my postdoc. And so what's pretty interesting is across Stellar's jays, there's this pretty deep split. And so if you can read that number that says 7.79%, that's how divergent they are in their DNA. So as a perspective, we share about 99% more even of our DNA with bonobos and chimpanzees. So these are quite divergent genetically. And so there appears to be this split between the Rocky Mountain and Southern group of Stellar's jay from the Pacific lineage of Stellar's jays. And so they do differ in their morphology as well in terms of coloration on their frontal stripes, as well as their ecology too. And so we suspect and will propose in the nearest future, this is yet to be published, but there will probably be a split involving Stellar's jay in the future. So one of the other things we're interested in is biogeography. And so that's trying to understand the geographic distributions of birds in the context of their evolutionary biology. And so for this, I want to briefly touch on some work that I've done with this group of birds called the seed eaters. And so again, this is a bird that makes it barely to South Texas. I think this is the only actual tanager, so true tanager from the family Thrauphidae you can actually get in North America, or sorry, I should say the United States rather, and it occurs just in the southern tip of Texas. When we started this project, this was a group where we had 3 subspecies, and so there's the cinnamon rump seed eater, which occurs in red, and then there's also the white-collared seed eater, which occurs here in blue, and then a subspecies of another one, Sharpei, which is in yellow, the one that we get in Texas occasionally. And so again, we sequenced DNA from these different populations, but we actually found a very different pattern from what we saw in the redpolls. What we saw, and this is something called a structure plot, but this is a way that we visualize population assignments based on genetic data. So the fact that these are different colors tells us that these are different genetically from each other. And so what we find is that these lineages are actually quite divergent from each other. So the Pacific Coast one, the cinnamon rump one is very different from the ones that are on the Caribbean. And if we actually look at the DNA of these 2 populations that are supposed to be the same species and compare them to what we see across the whole genus, we actually see that they're really distantly related to each other. And so again, this is a phylogeny. And so here is the Pacific coastal one, and here is the Caribbean one. And you can see that they're actually far apart in the phylogeny. So what this appears to be is an instance where there's similarity in plumage. And so these look pretty similar, right, in terms of their overall patterning, but that's probably just convergence or the independent evolution of this plumage pattern. And they're actually very divergent, different species. And so these have now been split. So we have a cinnamon rump seed eater here, which is now Sprophalotorquilla, and the Sporophila morleti is the white-collared, or morlets seed eater is what it's called now. But this also kind of helped us think about the biogeography of this group, right? So if they're both on the northern part of this collective distribution, so all of this green that's shown here is the distribution of the entire genus, this new information that we have about the relationships changes understanding of when and how many times this lineage has colonized Mexico from Central and South America. So briefly, I just want to talk about an exciting ongoing project that I have that I alluded to that I'm excited to pursue during my first few years here at LSU, and that's focusing on this ecosystem that are called the polylepis in the world in terms of elevation and occurs throughout the Andes Mountains. And so it occurs as these stunted trees. I just got a message on my internet is unstable. Hopefully it's okay, but we'll continue going. So it's these stunted trees here, and the distribution is in the little blue dots that are shown here. And so basically they're these islands of habitat that rise out of grasslands, and they're endangered by habitat loss and climate change, but there's bird communities that specialize on these little islands of habitat. And so here's some of those species that occur in these polylepis islands, and I'm hoping to go down to this region and basically study their evolutionary biology and their population genetics to understand connectivity among the different islands of habitat and how that might differ among these species. And so these species vary in terms of how specialized they are. So some of them are highly specialized, like this giant conebill that only occurs in the polylepis forest. Other ones are more moderately specialized. They occur in the polylepis, but they also occur in adjacent habitat. And so this comparison gives us really interesting framework to think about the evolutionary biology of the bird community that occupies this endangered ecosystem. Okay, so I'm going to talk a little bit about some of the work that I've done on bird song and bird color next. So a lot of the work I've done is focused on tanagers. I've already mentioned tanagers a few times, and not the Paranga tanagers, so not the scarlet and summer tanagers that we get here in North America, but the tanagers in the family Thraupidae, which are a neotropical family of about 370 species. So this was the group that I sort of that I gained my chops with as an ornithologist. I studied them during my master's degree, and specifically I focused on their vocal evolution. And so in addition to being a very diverse group in terms of their color and their ecology and their natural history, they're also very diverse in terms of the vocalizations that they make. And so the first projects that I did when I got interested in ornithology was analyzing a whole bunch of different bird songs from these different tanagers. And so you may be familiar with these representations of song. They're called spectrograms. There's frequency on the y-axis and time on the x-axis. And so with this information, you can actually get a lot of characters related to, well, how long is the song? How rapidly does the bird sing? What frequency does the bird sing at? And so we amassed this really large data set, almost 3,000 songs across over 300 species, and got all of these different characters related to things like the length of the notes and how much bandwidth the notes spread across. And then from that, we could test a bunch of hypotheses about how songs evolve. So the one thing I'll share with you now is this idea of what's called the acoustic adaptation hypothesis. And you may have heard of this. It's the idea that vocal evolution is optimized for transmittance in different habitats. And so if you think about a forest versus a grassland area, they're very different in terms of it as an acoustic space. And so evolution may favor vocalizations that are better transmitted across different environments. And specifically, if you think about frequency as a waveform, or sound as a waveform, we would expect in denser habitats in forest for birds that have lower frequencies to better transmit their song than those that have higher frequencies. And so we can make a bunch of predictions in terms of both the temporal and the frequency related aspects of song to try figure out how song has evolved in this group in relation to habitat. And science is never, it's not always as clean as you might initially hope, and so we actually found mixed support. So some of the characters that we studied did support this idea that there are predictable differences between open, so like grassland and forest habitats. So things like frequency shift rates, so there were more really rapid changes in frequency in grassland habitats as opposed to forest habitats. But other things like peak frequency did not appear to match this prediction that we expected. And it's important to keep in mind that this is across 300 species, and so in some instances we actually do see examples of this within a species or among closely related species, but these were the general trends that we found. And so in addition to the work I've done with song, I've also done a lot of work on color. And so specifically, a lot of my current work and previous work has focused on horned larks. And so horned larks are a really fascinating lineage of birds. I feel like little brown jobs sometimes get they get a bad reputation, but I think they're really fascinating in terms of their biology. And so we have horned larks here as the sole representative of the family Alaudidae. So larks are a family of about 100 species with mostly distribution in Asia as well as Africa. But horned larks currently have this really remarkably wide distribution, right? So there are the same species of horned lark we have in Northern Africa, as well as in Mongolia, as well as Northern Russia. And so they occupy all of these different habitats, from tundras to deserts to grasslands. And so I'm interested in trying to understand how they're able to occupy all of these different environments. And if you've ever, so here, these stars indicate some places that I've been able to go to, been fortunate to work in to study horned larks. And I'm often envious of some of my colleagues' field site photos, because for horned larks, if you know horned larks, sometimes they like dirt habitat, dirt patches. Sort of sometimes they're parking lot birds. So here are some of the places that I've seen. But you can see they tend to prefer open areas with little to no vegetation. So this is in Toca, Colombia, where we found some. This was in Brawley, California, and I'll come back to this population actually. You can see just a dirt field in agricultural land, and then again in Oaxaca, Mexico. So you're starting to see a trend here in terms of horned lark habitat. But I'm as interested in thinking about these different habitat and substrates from the context of camouflage. And so if you've ever observed horned larks, sometimes you can hear them and they're there, but they can be difficult to find. And so I was interested in quantifying camouflage in horned larks across their range. So this is work I did as a postdoc, and what was really fascinating about horned larks is they have all of this geographic variation in the western United States. So each of these different colors corresponds to different subspecies of horned lark, and that really corresponds to differences in dorsal coloration and patterning, as well as some facial patterning and body size. But there's this amazing collection at the Berkeley Museum where I did my postdoc. I took photographs of these birds using a special camera that's been modified to capture UV as well, so birds can see UV. And so basically you can draw these polygons and also get information on patterning. And so hopefully this animation will play. But what you can do is you can actually quantify and identify sort of the dark spots on top of the lighter feathers. And you can quantify changes in patterning as well as the base color among the birds using digital photography. And then you can compare that to environmental variables. And so we have information on soil data sets. So there's been remarkable advances in the availability of data in terms of GIS and geographic information systems, and that's made some really interesting comparisons available. Specifically, I was interested in quantifying organic carbon and calcium carbonate in the soil as a potential way to quantify color. And in addition to color, I was also interested in the composition of the soil with respect to the particle size. And so you can actually get data on how much sand is in the soil versus how much silt and how much clay. And so we have a phenotypic data set on the appearances of these horned larks, and we have environmental data. And we combined them, and we found that, yes, in fact, there is background matching in terms of the brightness. And so larks that are brighter tend to occur on brighter soils. And larks that are redder, have higher achieved chroma, tend to occur on redder soils, and also in terms of patterning as well. And so birds that have more back patterning tend to occur in soils that have more sand. And so what seems intuitive, if you just think about the horned lark as a bird in its natural history, we're actually able to quantify how it has changed across this entire range using museum specimens. And so this will parlay into the next bit, which is still on horned larks, but think about, so that was across space, but what about across time? And so what happens if these habitats change over time? And so if you think about it, populations only have a few options. They can either adapt or acclimate if they have, basically, so they can adapt. Adaptation happens over multiple generations, or if an individual can change its appearance, they can do that, which would be called acclimating. They can move. They can go to a different place where they have a better suited appearance or adaptation for that environment, or they might go extinct. So one of the places that I've done work with horned larks is the Imperial Valley in California. And so if you've ever been to southeastern California, you'll know that it's a pretty remarkable place. The Salton Sea is a highly sought after birding destination, nice place for seeing migrant birds. And so, yeah, so there it is in southeastern California. But it's a fascinating place because there's been this huge advent of agriculture. And so historically it was desert flats, it was sort of mesquite, creosote bush. But now with the construction of the All-American Canal, which then burst and created the Salton Sea, as well as all this agricultural land, there's been dramatic changes to this environment. And so ornithologists collected a series of larks from this area in the early 1900s. And so we're able to have this historical series of birds that we can compare. And so we went back and collected additional specimens and found that they are in fact darker. And so you can see there's darker dorsal plumage on these birds that were collected more recently. And so it seems like there's been a shift in the lark appearance over time as agriculture has been developed in this region. And so it seems like contemporary larks are darker than the historical larks are. And I won't get into this, but we also use other things to analyze their diet and how their diet has been shifting as agriculture has moved into the area. And so they're incorporating both food and water from this foreign source of agriculture. Okay, I'm running out of time, but I just briefly want to talk about Louisiana and some of the work that I'm doing locally and hoping to do. So this is stuff that I'm just getting started. But one of the really interesting things about museums, and that I think is vitally important about museums to think about, is well put here by one of my personal heroes, Joseph Grinnell. So he was the founder of the Museum of Vertebrate Zoology at Berkeley. And he says that the greatest value of our museum will not be realized until many years when the student of the future will have access to the original record of faunal conditions wherever they work. And so if you think about natural history museums, they provide these snapshots of what nature was like in that place in that time. And so one of the things that we're hoping to do is to improve our collections locally. And so while I've been telling you about how amazing our collection is across the world, there's actually still a lot of work to be done locally in terms of understanding Louisiana avifauna. And so this is a map showing the distribution of specimens that we have in our collection. You'll notice that the whole state is not actually covered. There's actually some parishes for which we have essentially no specimens. Also, this is across all seasons. So there are relatively few breeding birds from Louisiana. And so I'm hoping to improve our series of Louisiana birds so we can track change in appearances like I did with the larks. But also there's pretty amazing things you can do to look at changes in diet just from the feathers, as well as exposure to different pollutants. And so pollution is a serious, important issue here in Louisiana. And we can actually use historical series and spatial series of specimens to understand the fate of pollutants in our ecosystems using museum collections of birds. So one of the other things we do is we collaborate with other folks opportunistically to try to understand ways in which birds are being affected by different events. And so one of the things that we've been involved with is during the snowstorm. I mentioned this when I was talking to some folks before our talk started, but I didn't think it snowed in Louisiana. I grew up in Minnesota, and so I was really surprised when I moved down here and experienced that crazy snowstorm last year. And it took a lot of purple martins off guard too. And so there was a huge die-off of purple martins. And many of those specimens we've actually been able to collect and salvage. So we have an ongoing study that's collaborative where we're actually trying to look at what happened physiologically to these purple martins and try to understand how they were impacted. And so these are the kind of things that we also do opportunistically to try to understand impacts on birds. All right, so just very, very briefly, I want to talk about two things for the future of the museum. So historically, museum-based ornithology has been really dominated by white men, frankly. And so one of the things that we're trying to improve in the museum is our way in which we are being more diverse and more inclusive for other groups. And so one of the really exciting things that's happened recently, it was the Emily Sneffledge expedition, which was led by a majority of women. This is the first time this has really happened. This is a really awesome group of women that went down to Brazil in the Amazon and led this expedition. Glaucia Del Rio here and some other folks that I've mentioned were part of this. And so we're hoping to diversify the museum community through outreach and basically education as well, and providing opportunities to folks that have been historically underrepresented in museum communities. And I briefly referenced this, but one of the exciting things that's actually going on in the museum is there's some pretty big shifts in terms of space. And so if you go back and read the Eyes of the World link that I put at the very beginning, that's all about the beginning of the museum. There was this one part where Lowry, he is forlorn, he's really sad because he can't finish the public exhibits, because basically, if you've ever been to Foster Hall, where a museum is housed, we share it with the art department. And that's been that way since the 1930s. And so we were worried for decades that without buy-in from the university, we were going to basically run out of space, and this was going to really hamper our activities. But I'm happy to report that there's a new studio art building on the south end of campus. What this means is that there will be an increase in the amount of space that's available for growth of the museum, and this is a really, really huge deal for us. And George Lowry would be thrilled if he heard about this. And so this is a really exciting development. I think the space is turning over to us in 2023 in the spring, but this is going to be a really long, ongoing project to try to maximize how we use this space. But it's an exciting new chapter for the museum. So I just really want to end by saying there's various ways that you can support the museum. You can follow us on social media. We do a lot of outreach events. There's ways you can support student research. We do a big day every year, often in the tropics, where we'll see hundreds of species, and we've broken records in the past in terms of the number of species observed. And we're going to have certainly a lot of fundraising and outreach initiatives related to Foster Hall in terms of this new chapter. But more generally, we are, many of us, all of us are naturalists and bird enthusiasts in general. So, join our mission collectively to try to help us understand and conserve nature. And I'm going to just drop some links for the social media in the chat, if that's okay. So you can follow us if you're interested. And so that's it. I just wanted to say thank you again. Sorry I went a little bit over time, but I appreciate the opportunity to speak with all of you, and look forward to hopefully meeting some of you in person and going out birding someday soon. And I'd be happy to take any questions.